Hi, my today's talk about computer-aided drug design. And it will consist of two parts. First, I will introduce drug discovery in general, the process, deliverables, and technologies used. Next, I will provide a more detailed view of how computers help to discover new drugs with specific examples, mainly from my lab, but also a few cutting-edge studies by other groups. But before I get into details about the discovery process, I have to mention three premises, or should I say three central dogmas of modern drug discovery. The first one is that a disease, in general, is caused by a perturbed function of a protein called target. Modulating the target by a chemical compound, drug, may cure the disease. This makes of pharma companies a major consumer of latest advances in biomedical research. The second is that a new drug must be safe for intended use, that is, provide greatest therapeutic benefit without resulting in unacceptable side effects or toxicity, which makes drug development a long, highly regulated, and risky endeavor. And finally, drug discovery is a costly enterprise. A drug molecule must be covered by a patent so that the drug developer could protect its investment. And here is a big picture of a full discovery cycle. In most, if not all companies, there is a separation between two phases called discovery and development. Discovery starts with the target identification and eventually delivers a drug candidate. Development takes the candidate and moves it through preclinical and clinical phase 1, 2, and 3 trials. The major difference between the two stages is that in discovery, new chemical compounds are made until a compound that checks all boxes on the candidate checklist is identified. In development, the drug candidate either passes all clinical phases or fails. A new candidate cannot be introduced in the middle of phase 2, for example. The full discovery and development cycle takes 8 to 15 years and costs on average $80 million, of which discovery weighs about a quarter in time, both in time and costs. Now, I'll briefly describe all discovery steps, though I'll exclude the target identification since it's usually being done as a separate publicly funded research. Then the first step would be identification of a lead compound, which starts with high throughput screening, or HTS. HTS delivered what is called hit compounds, that is, compounds showing weak or moderate activity. Medicinal and computational chemists analyze those hits to pick one that is most likely to become a drug. Such compound is called a lead. Next step is lead optimization. At this step, the medicinal chemists, with some help from computational designers, make new molecules that are tested against a broad panel of biochemical, biophysical, cellular, and in vivo assays until they obtain one that has an optimal profile against all of those assays, and in addition is able to reach its molecular target in vivo. Here I listed key technologies or technological platforms that shaped modern drug discovery as we know it today. So they include protein purification and production, X-ray crystallography, organic synthesis, multiple assay technologies, biophysical, biochemical, cell-based. Also important is assay miniaturization, compound library management, and creating or development of transgenic organisms. I also felt like I should mention a phenomenon of technological revolutions. You know, that moment when people say, from now on, things, the discovery will never be the same. And almost all of these technologies went through a well-described hype cycle that includes trigger, inflated expectations, disillusionment, and finally a plateau of productivity. So currently, almost all of these listed technologies are at the plateau of productivity, maybe except two latest that I highlighted in green, which are development of new class of drugs called protein degraders and artificial intelligence, and in which, well, protein degraders, it's more like industry specific thing, and artificial development is a, I would say, global phenomenon. In the next couple of slides, I'll describe a typical drug discovery project from our own practice started in 2009. Of course, I'm not sure if such thing as a typical drug discovery project even exists. Here, we targeted not one, but three targets, a family of receptor tyrosine kinases, tyro3, AXL and MER, or simply TAM. Two of them, MER and AXL, are ectopically expressed in multiple tumors and have been shown to promote cancer cell survival. These targets have been discovered by our collaborators, oncologists Shelley Erb, UNC Chapel Hill, and Doug Graham from Emory. Then our team, Stephen Fry and Xiaodong Wang, medicinal chemists, Bill Jensen, screening scientist, and myself, started structure-based design from scratch. Xiaodong has proposed a hinge binding scaffold. I docked it, and we came up with a few options for the scaffold decoration. The chemists started their synthetic work, and in a couple of months, after a few iterations of synthesis, screening, and design, we obtained a low nanomolar range inhibitor that we considered a lead. This is an extremely brief overview of the lead optimization project. In general, it's a winding road, like the green arrow, rather than a straight line through the three compounds shown. There are many lead qualities, such as in vivo activity, that are hard to rationalize and need to be improved through trial and error. Anyway, by 2015, we finally delivered a drug candidate that is currently in phase 1 clinical trial. And now I'm finally approaching the long-promised story about computer-aided drug design. There are many ways to classify a great variety of computational techniques. For instance, one clear distinction is between structure-based and ligand-based strategies. Here, structure-based means that three-dimensional structure of the protein target is used to drive lead optimization. In the ligand-based universe, the structure of the target is usually unknown, and new, better leads are predicted based on the structure and activity of other ligands. Also, most technique can be used in a predictive or design mode. Like I previously said, the main input to structure-based design is the 3D structure of the protein of interest. Even better when a crystal structure of a ligand-protein complex is available. However, a ligand can also be computationally docked into the protein. The structure of the ligand-protein complex is then analyzed on the presence of favorable and unfavorable interactions, and incremental structural changes to the ligand are proposed in order to eliminate unfavorable interactions while preserving favorable interactions. The main challenge here is to comprehend the entropic component to the ligand protein binding. Quantitatively, it can only be characterized by observing ligand protein complex in motion. However, observation of a static structure can also provide some useful hints. Here is an example of a structure-based study from my group. Here we used the novel original approach to assemble ligand from fragments directly in the protein binding site. Unlike most computational techniques that are virtual screeners, phrase-based design is rather a virtual medicinal chemist. The new concept was used to design a new chemical series of TAM inhibitors with better intra-TAM and kinome-wide selectivity by exploiting yet untapped protein pockets. The approach was validated by X-ray crystallography and proteomic studies, and new compounds showed high potency in cellular and in vivo assays. Probably the most popular branch of computer-aided drug design is structure-based virtual screening. In virtual screening, ligands from a large database are being docked one by one into a protein target. Docking is an algorithm to determine the ligand binding pose. It also includes scoring, that is a very approximate estimate of the ligand protein binding free energy. One particular strength of virtual screening is that it allows you to screen millions of compounds that are not available in your lab. Virtual hits can then be purchased and tested. It is important to remember that virtual screening is a very uncertain exercise with an extremely high false positive rate. The goal for a virtual screener is to obtain a set of virtual hits that would be enriched in true hits compared to a randomly selected set. But it is not expected to be exclusively composed of true hits. Typical success rates, that is percentage of true hits among selected compounds, is just a few percent. Here is a published virtual screening story from my group. Again, the target was Mer kinase domain. We screened a commercial library of approximately 4 million compounds. And in addition to the standard scoring function, also applied our original approach called structural protein ligand interaction fingerprints, or SPLEEF, to rank virtual hits. Eventually, 62 compounds were purchased and experimentally tested. 15 of them showed significant activity. One of them showed sub-micromolar potency that is quite rare for virtual screening hits. We also determined that the enrichment rate in true active in our virtual hit set was approximately 200-fold compared to a random set. I should also mention molecular mechanics and molecular dynamics as important research tools in computer-aided drug design. Molecular mechanics is used as a fast and affordable technique to determine 3D molecular structure. It can be applied to molecular systems composed of millions of atoms. To achieve this performance, empirical potentials of interatomic interactions called force field are used instead of solving Schrödinger equation for the whole system, which would be impossible for systems of interest in drug design. Molecular dynamics then combines molecular mechanics force field and equations of motion to simulate the movements and conformational transformations of the molecular structure. For example, in this study, we use molecular dynamics to help chemical biologists to design an allosteric agonist of a large epigenetic complex, PRC2, having a methyltransferase activity. Previously, only antagonists of this complex were known. Observing the dynamics of the complex, we noticed that motions and transformations of the structural element called SRM helix may be responsible for the activity or inactivity of the complex. We proposed that compounds that would stabilize the SRM helix would promote the PRC2 catalytic activity. This hypothesis was then confirmed experimentally. An important family of molecular dynamics techniques is being developed with an ultimate goal of theoretically rigorous and accurate predictions of the ligand-protein binding affinity. These techniques are collectively called free energy perturbation techniques or alchemical techniques. The term alchemical here is used since molecular alchemical term here is used since key feature of these methods is calculation of binding free energy differences between chemically impossible transition states of the ligands. One latest example of a massive use of such alchemical calculations is collaboration between several computational and medicinal chemistry labs called COVID Moonshot. In this project, people all over the world are invited to submit chemically tractable chemical structures of putative inhibitors of the SARS-CoV-2 main protease. The binding affinity of those crowdsourced compounds is then calculated by an alchemical technique on the computing platform called Folding at Home. And compounds with the highest predicted affinities are being synthesized and experimentally tested on their main protease activity. It's obvious that my overview would be ostensibly incomplete if I wouldn't mention ligand-based approaches. In the ligand-based realm, the goal is to predict biological activities for compounds based on their similarity or dissimilarity to compounds with known activity data. Many hundreds, if not thousands, ligand-based techniques exist. The major difference between them is how chemical structure is represented in the computer and how relationship between those representations is calculated. For example, in QSAR, which is probably the oldest branch of ligand-based phylogenetic tree, structures are represented as large sets of topological invariants calculated from molecular graphs, and then machine learning techniques are used to train a mathematical model that would be able to distinguish between active and inactive molecules. A very different approach consists in taking a 3D representation of an active molecule and hypothesizing which functional and spatial features are responsible for its activity. Such hypotheses, called pharmacophores, can then be used for virtual screening of large compound libraries. I thought I'd give this example of ligand-based design since it's probably the only QSAR paper published in Nature, well, at least in the last 20 years or so. Here, Andrew Hopkins' group at University of Dundee developed and applied an automated workflow to develop QSAR models for multiple GPCR targets and used them to evolve an approved acetylcholinesterase inhibitor drug into brain penetrable ligands with the specific polypharmacology profiles for G protein coupled receptors. The ligands so designed were confirmed and characterized in a broad panel of in vitro assays and in an in vivo model. In conclusion, I need to mention the newest kid on the block, artificial intelligence, which in general consists of application of deep machine learning techniques to various problems of computer-aided drug design, and more generally, computational chemistry. The most obvious use is probably in QSAR, where previously used machine learning techniques are simply replaced by deep neural networks, often even without scrutinizing whether the change results in any added value. And in less obvious cases, deep learning is used to generate, for example, accurate solutions for Schrödinger equation. To illustrate AI use in drug discovery, I picked this paper in Science Advances from Alex Tropsche group at UNC Chapel Hill. Here, the authors devised a new strategy for de novo design of molecules with desired properties termed RELEASE, that stands for reinforcement learning for structural evolution. RELEASE integrates two types of neural network, a generative and predictive one. They were trained separately, but then used jointly to generate novel targeted chemical libraries. As a proof of concept, RELEASE was used to design chemical libraries with a bias towards structural complexity, hydrophobicity, and inhibitory activity against Janus protein kinase 2. And in the end, I wanted to provide a few perspectives of where computer-aided drug design and drug discovery in general probably must be going in the next few years. And so first of all, I would say we will need to dramatically increase the pace and cut the costs of developing probes and drugs for new targets. So in fact, there are an estimated 3 to 10,000 disease-causing proteins that might be targeted by small molecule drugs for which no ligand have been developed yet. At the current pace, the goal of having small molecule probe for every target would take decades and tens of billions of dollars to accomplish. And hence, new computational strategies are needed to accelerate the process. Also, we will have to be able to make predictions for compound potencies in animal models and humans to decrease the drug attrition rates, and which can probably be done by connecting genomics data to chemistry and to clinical data. And that is all for today. Thanks for listening.